DNA is often called the code of life because it allows all living organisms to store and read information which allows organisms to perform the functions necessary for life. And crucially, it also allows the transfer of this information from one generation to the next, ensuring that successive generations inherit the instructions for life from their predecessors. DNA is found in the nuclei of all living cells. However, from grade 10, you might remember that the nucleus contains the chromatin network, or chromosomes, which does make the concept a bit confusing. So let's clarify the differences between chromatin, chromosomes, and DNA. The nucleus contains long thread-like structures called chromatin. This chromatin is made up of DNA, which is wrapped around proteins called histones. So in fact, when we say that DNA is found within the nucleus, we are referring to the DNA that makes up the chromatin network. In preparation for cell division, DNA has to replicate, which means it makes a copy of itself, which also essentially means that the amount of chromatin, or the chromatin network in the nucleus, doubles. And then during cell division, the chromatin condenses to form structures called chromosomes. These chromosomes will then split the genetic material between the two cells, so that one copy of DNA goes to the one cell and the other copy of DNA goes to the other cell. We've discussed cell division before when we looked at mitosis in grade 10, and we will discuss another form of cell division called meiosis in a later topic. However, for now, it's important that you understand the difference between DNA, chromatin, and chromosomes, and when we refer to the different structures. For the remainder of this topic, we are going to focus on DNA and other nucleic acids which are found within the nucleus and the rest of the cell. But first, what are nucleic acids? Nucleic acids are organic molecules. And if you remember from grade 10, organic molecules are molecules that are made within the cells of living organisms. Nucleic acids have two very important functions. They control the synthesis of proteins, which means they control the process of making proteins. They also store and transfer hereditary information. Hereditary information is the information that is passed on from one generation to the next and determines the characteristics of organisms. There are two types of nucleic acids. DNA, as we've discussed, and the full name for DNA is deoxyribonucleic acids, and RNA, or ribonucleic acids. First, we will look at DNA, starting with where in the cell will we find DNA. DNA is found mostly in the nucleus, and this is also where most of the processes we will look at in the rest of this topic, which involves DNA, will take place. However, some DNA is found within the mitochondria of cells, called mtDNA, and plant cells also have DNA within their chloroplasts. But as mentioned, most of the DNA is found in the nucleus, and that's also where most of our focus will be. Looking at the structure of DNA, we see that DNA consists of two strands which are twisted to form a double helix shape. If you look at the diagram, you can see the double-stranded nature of DNA as well as the twisted nature of DNA, which gives it that characteristic double helix shape. DNA consists of monomers called nucleotides. Monomers are the building blocks of molecules. So the building blocks or the monomers of DNA are called DNA nucleotides. The structure of DNA was discovered by various scientists in the 1950s. The first scientist to lay eyes on DNA was Rosalind Franklin, who took an X-ray image of DNA in 1952. She then showed this image to a colleague called Maurice Wilkins, who showed that image to two other colleagues called James Watson and Francis Crick. who then went on to formulate the double helix structure of DNA. Almost a decade later, Watson, Crick, and Wilkins received a Nobel Prize for the discovery of DNA. Notably, Rosalind Franklin was excluded from receiving this Nobel Prize. Taking a closer look at the monomers of DNA, or DNA nucleotides, we can understand the structure of DNA a bit better. Each nucleotide consists of a sugar molecule called deoxyribose, as well as a phosphate group and a nitrogenous base. There are 4 different nitrogenous bases, which then gives us 4 different nucleotides. And these 4 different nucleotides makes up the structure of DNA. The first nitrogenous base is adenine, then guanine, cytosine, and thymine. We use the letters A, G, C, and T to depict the 4 different nucleotides that make up DNA. Using the 4 different nucleotides, DNA can form molecules which are endlessly varied and makes all of the complexities of life and differences in living organisms possible. The deoxyribose of one nucleotide binds with the phosphate group of another. This then forms 2 long strands of nucleotides. The 2 strands bind together via weak hydrogen bonds. The weak hydrogen bonds forms between complementary nucleotides. The complementary nucleotides in DNA is A and T and G and C. This means that A, or adenine, always binds with T, or thymine, whereas guanine, or G, always binds with C, or cytosine. This will become a bit more clear later on when we discuss the different processes that involves DNA. But what is important to remember is that A always binds with T and G always binds with C. This also means that the number of adenines in a DNA molecule is always equal to the number of thymines, and the guanines are always equal to the number of cytosines. And finally, we're going to look at the functions or the importance of DNA. DNA carries the hereditary information within the cell, meaning the information that gets passed from one generation to the next. We're going to learn more about the hereditary information in a future topic when we look at genetics. DNA also contains the coded information for protein synthesis. And later on in this topic, we're going to look at the process of protein synthesis, and you will understand this point a bit better. But it's important to understand that the information for protein synthesis is coded within the DNA found in the nucleus. That then brings us to the other type of nucleic acids, RNA, or ribonucleic acids. RNA, similar to DNA, is found within the nucleus. However, unlike DNA, RNA is also found within the cytoplasm. The structure of RNA is also different from DNA. DNA had a double-stranded structure, whereas RNA is single-stranded. And even though this diagram depicts RNA as having a helical shape, that isn't necessarily the case. RNA has no particular shape, or we can say that it has a straight shape. Important is RNA also consists of nucleotides. However, the monomers of RNA are called RNA nucleotides because the nucleotides of RNA is slightly different than that of the DNA nucleotides. Speaking of which, let's look at RNA nucleotides. RNA nucleotides consist of a sugar molecule called ribose, unlike DNA, which consisted of a sugar molecule called deoxyribose. RNA nucleotides also have a phosphate group, similar to DNA, and it also has nitrogenous bases, similar to DNA. However, there's one key difference in the nitrogenous bases that makes up RNA nucleotides. The nitrogenous bases of RNA nucleotides are adenine, guanine, and cytosine, which is similar to DNA nucleotides. However, the fourth nitrogenous base of RNA nucleotides is uracil, which is the key difference between DNA nucleotides and RNA nucleotides. RNA forms when the ribose of one nucleotide binds with the phosphate group of another nucleotide. This forms a long single-stranded molecule, unlike DNA, which has a double-stranded molecule. This also means that the nitrogenous bases of RNA can occur in any ratio. So the nitrogenous bases of adenine, uracil, guanine, and cytosine occur in any ratio, and there is no particular rule that indicates that certain nitrogenous bases have to be equal in number. Unlike DNA, where the nitrogenous bases of adenine and thymine occur in equal numbers, and the nitrogenous bases of guanine and cytosine occur in equal numbers. The function of RNA is its role in protein synthesis, which we will look at a bit later on when we look at the process of protein synthesis. But it has two very important roles in protein synthesis. There are a couple of distinctly different types of RNA. We're going to look at two of these types of RNA specifically as they relate to their role in protein synthesis. The first type of RNA is messenger RNA, or mRNA. mRNA is formed or transcribed from the DNA in the nucleus, and it then carries the message for protein synthesis from the nucleus to the ribosomes in the cytoplasm. Transfer RNA, or tRNA, has a folded single-stranded structure, and its function is to carry amino acids in the cytoplasm and bring those amino acids in sequence for protein synthesis. We will once again discuss this process of protein synthesis a bit later on. So if we were to look at the similarities and differences of DNA and RNA, we should be able to come up with the following list. Both DNA and RNA are nucleic acids. They both have a phosphate group. They both have nitrogenous bases of adenine, guanine, and cytosine, and they both carry genetic information. They are also both involved in protein synthesis. That then allows us to look at the differences between DNA and RNA. DNA has a double-stranded structure, whereas RNA has a single-stranded structure. DNA has a helical shape, whereas RNA has a straight shape. The nucleotides of DNA consist of a sugar molecule called deoxyribose, whereas RNA nucleotides have a sugar molecule of ribose. One of the nitrogenous bases of DNA is thymine, whereas uracil is a nitrogenous base found within RNA nucleotides. The bases of DNA are paired, meaning that the number of adenines is equal to the number of thymines because they have to bind in the double-stranded structure of DNA, whereas there is no pairing of bases in RNA. Now that we've looked at the basic structure of DNA and RNA, we can look at some of the processes that involve these nucleic acids. The first process we're going to look at is DNA replication. DNA replication is the duplication or copying of DNA. This happens in preparation for cell division, where the cell needs to duplicate its DNA to form two identical copies. This ensures that each daughter cell has the identical genetic material to the original cell. It takes place in the nucleus during interphase in the cell cycle. Let's look at the process of DNA replication. As mentioned, DNA takes place within the nucleus of the cells, where the DNA is found. The first thing that needs to happen in order for DNA replication to take place is DNA needs to unwind. This then allows the weak hydrogen bonds between the nitrogenous bases to break, or we say that the DNA unzips. This then forms two separate strands, each of which will serve as a template for building a new DNA molecule. Free DNA nucleotides from the nucleoplasm will then bind with complementary bases on each of the template strands, resulting in two complementary strands forming on each template. So adenine binds with thymine and guanine binds with cytosine. This then forms two identical DNA molecules, each of which has one original strand and one new strand. If you compare the two molecules at the end, you will see that they are exactly identical. The process of DNA replication is controlled by various enzymes. You do not need to know the specifics about the roles of these enzymes, but it's important to mention that enzymes are involved in the process. So why is it important for DNA replication to take place? Well, as mentioned, DNA replication takes place in preparation for cell division to ensure that each cell has identical DNA to the original cell. In mitosis, DNA needs to be doubled to ensure that each cell receives the same amount of DNA to the original cell and that each daughter cell is genetically identical. For meiosis, DNA needs to be doubled to ensure that 4 haploid cells can be produced with the same amount of DNA, and that the genetic material is transferred from parent to the offspring. That then finally brings us to the process of protein synthesis, as alluded to throughout this video. It is one of the most important processes that takes place within the cells of living organisms, and we're going to look at exactly how that process takes place. Firstly, we need to recap what exactly is a protein. From grade 10, you'd remember that proteins are organic compounds, and they are long chains of amino acids that are held together by peptide bonds. So the monomers of proteins are amino acids. Protein synthesis is the process whereby proteins are manufactured within living cells. And DNA and RNA controls the process of protein synthesis. We've mentioned that DNA contains the coded message for protein synthesis. However, one molecule of DNA contains the coded message for many proteins. So a segment of DNA that codes for a specific protein is called a gene. The coded message for protein synthesis is provided by the sequence of nitrogenous bases. 3 consecutive nitrogenous bases codes for a specific amino acid to be added to the protein molecule. We call these 3 consecutive nitrogenous bases a base triplet. So the sequence of base triplets within the gene will tell us the sequence of amino acids that needs to be added in order to make a particular protein. The process of protein synthesis occurs in 2 distinct phases. The first phase is transcription, which takes place in the nucleus. This is where the coded message for protein synthesis from the DNA is copied onto an mRNA molecule, because DNA cannot leave the nucleus, so it needs to copy that message onto an mRNA molecule, which can then leave the nucleus and go to the cytoplasm, where the second phase can take place, called translation. This happens at ribosomes in the cytoplasm, where the protein molecule will be built. The first phase of protein synthesis, transcription, takes place within the nucleus of the cells. This is where the DNA molecules containing the coded message for protein synthesis is found. Firstly, the DNA unwinds. Then the DNA unzips, meaning the weak hydrogen bonds that hold the 2 strands together breaks, allowing 2 separate strands to form. Only one of these strands will form the template where the mRNA molecule will be built. 3 consecutive nitrogenous bases on the template strand is called a base triplet. Free nucleotides from the nucleoplasm will then start building the mRNA molecule on the template strand of the DNA. 3 consecutive bases on the mRNA is called a codon. The mRNA now contains the coded message for protein synthesis and can leave the nucleus via the nucleopores and enter the cytoplasm, where it can take that message to the ribosomes, where the building of the protein will take place. The next phase of protein synthesis, translation, occurs in the cytoplasm at organelles called ribosomes. The mRNA molecule that was formed during transcription arrives and attaches to the ribosome, carrying the coded message for protein synthesis. tRNA molecules are free-floating in the cytoplasm, and they carry specific amino acids. The specific amino acid of the tRNA is determined by 3 exposed nitrogenous bases on the tRNA molecule called an anticodon. When the anticodon on the tRNA matches the next codon on the mRNA, it brings the required amino acid to the ribosome. When these amino acids are brought in sequence, they can bind together via peptide bonds. This process is repeated until eventually the required protein is formed. When analyzing the structure of a protein or the sequence of amino acids involved in making a protein, we need to consider the 3 nucleic acids involved in the process of protein synthesis, namely DNA, mRNA, and tRNA. DNA provides us with the template strand, which contains the coded message for protein synthesis. 3 consecutive nitrogenous bases on the DNA template strand, which codes for a specific amino acid, is called a base triplet. The complementary mRNA strand that forms from the DNA template strand have 3 consecutive bases called a codon, which will code for the same amino acid. And the 3 exposed nitrogenous bases on the tRNA is called the anticodon and will carry the specific amino acid. When we are asked to analyze the amino acids involved in protein synthesis, we will be given a table as follows. And in this example, the table contains codons, but it might also contain anticodons or DNA-based triplets, so read carefully. In this specific example, we need to look at the mRNA molecule and the codons on the mRNA molecule. The codon in this case is CGU, which means that the amino acid coded for by this codon is arginine, which means the amino acid carried by this specific tRNA molecule is arginine. Lastly, we're going to look at DNA profiling, which is a technique used by forensic investigators and scientists to analyze the specific DNA that makes up a specific individual. DNA profiling involves taking samples of DNA and processing them so that they can be represented as lines which correspond with a sequence of base pairs. This is called a DNA profile or a DNA fingerprint or barcode. We use DNA profiling to help us identify crime suspects, to show proof of paternity, to identify relatives, to determine the probability of genetic defects, and to establish the suitability of organ transplants. However, DNA profiling does not come without its issues. Some people believe that DNA profiles can be used to frame someone, where DNA can be planted to make someone look guilty in a crime investigation. It also leaves itself open to possible human error, which could give incorrect results. And lastly, a lot of people are uncomfortable with the possible invasion of privacy brought about by DNA profiling. Despite that, DNA profiling still has many useful applications. One such example is proof of paternity. A child's DNA makeup is entirely determined by the DNA of the mother and the DNA of the father. Meaning if we were to look at the child's DNA profile and we eliminate all the bars that correspond with the mother, the remaining bars must belong to the father. So if we look at this example, the first bar corresponds with the DNA profile of dad2 and dad3, as well as the last bar corresponds with both dad2 and dad3. However, the remaining bar is only present on the DNA barcode of dad3, which means dad3 is the father of this child. In the next example, we're going to look at how DNA profiling can be used to identify the suspect in a crime investigation. In this example, a piece of evidence was found on the crime scene which had some DNA on it. However, some of the DNA will correspond with the DNA of the victim, so we must eliminate that DNA from the DNA profile first. The remaining DNA barcodes must then belong to that of the suspect. So if we look at the first bar, it could belong to either suspect 1 or suspect 2. However, the second bar only belongs to suspect 2, which eliminates suspect 1 from the investigation. However, the third bar must also be present, otherwise that eliminates suspect 2. But in this case, the third bar does belong to suspect 2, which means the suspect in this investigation would be suspect 2. And that's it for the topic of DNA. I hope that you found this video useful and that you now have a better idea of some of the key concepts in this topic. Good luck with the rest of your preparations, and I recommend that you make your own summaries of the content and go over as many past paper questions as possible, as the best way to learn is to do. See you guys in the next one.